and I did want to talk about microbiome stuff. Can you start off by just defining for people that don't know what the microbiome is and why it's such a big focus of research right now?
Sure. So the microbiome refers to essentially all the microbes that live in a particular environment. So one of the most common that we think about is the gut microbiome. So what this means is that in your body, in your gut, are basically tons of microbes that live there, reside there. They perform metabolic transformations and that are essential to a lot of what goes into making you healthy or not healthy. So it can incorporate. So the microbiome is really looking at all of these other living organisms that are on us and inside us. And it is really an important thing to understand because microbes are what first evolved, the life that first evolved on this planet. So the vast majority of life actually exists as microbial species. And so that is just the interface, so every interface that we have, whether it's our skin, our mouth, our gut, basically everything we have that interfaces with the outside world in some way is coated with these microbes.
Interesting. So with the gut microbiome, can you give people a sense for I've heard, and I don't know if this is true, that there's more microbes in your gut than there are cells in your body. What's the level of diversity and volume that we're talking about here?
Yeah, there was kind of a lot of analysis of that at one point, but basically, yes, if you look at the number of cells in your body and the number of microbial cells that you have in your gut microbiome, I believe that the latest numbers are that it's about equal. Yeah, initially it was thought to be about 10-1, and then people looked at it a little more and said, well, it's probably a little bit too much, but about the same number. So you've got a massive number of these. If you do the count differently, right? If you count, the total genetic content of your microbiome is vastly higher. I think it's 100-1. It's been a little while since I looked at that number, but I think it's about 100-1, the amount of kind of genetic content that you have in your microbiome compared to your human genome. So there is a huge amount of information that's there. And what we're finding is that it is incredibly relevant to health. And it is still very much a cutting edge area. And, we've done a number of studies in this, and I'm sure we'll talk about a few of them here in a second. But it is an area that people are just really trying to get their minds wrapped around and push out as much information as they possibly can. And this is, we actually have a gut microbiome test, so people can take that and they can go through, they can get a measurement of the microbiome, and it gives back a whole host of information about, relevant to health, both good signs and bad signs, and things you can do to make an impact on it.
Mm-hmm. I think let's first talk about what these microbes are doing and what we know about that, and then talk about things like the health impact and the variation between people and across lifespan. So in general, what exactly are these microbes doing in a physiological sense? Are they pre-digesting certain nutrients for us? Are they secreting things that are beneficial for the body or that we need for different things? What do we know there?
Yeah, it's all the things that you just talked about. So they do create certain compounds that have been associated with health. Butyrate, indoles, things like this have been shown to have positive health effects. They also make molecules that have a negative health effect. So one example of this is if you have certain species of bacteria, they will utilize material that they'll find in things like red meat, in eggs, and different aspects of the diet. And they create a compound called trimethylamine, or TMA for short. And then TMA will get into your bloodstream, and then your liver actually converts it into trimethylamine oxide, TMAO. And TMAO is a risk factor for cardiovascular disease. And so one of the things that happens there then is you can actually have compounds that in the absence of this bacteria are actually beneficial to you. But if you have those bacteria, it makes this TMA, creates the TMAO, in the liver, and you end up with problems from that. And so there's this whole network of metabolic transformations that are mediated by the microbiome for both good and bad. And I can give a couple other examples. So another one is that the microbiome actually interfaces with drugs we take as well.
Mm.
So there was a really fascinating study that came out in, I think it was in Cell earlier this year. And they went through and did a really deep dive on one particular individual's microbiome. And then they tested it against a whole host of drugs to see what happened when those drugs were exposed to the microbiome. And what it turned out was that 13% of the drugs were inactivated, right? They were just eaten or changed into some other substance or made inert or, whatever it was. So you inactivated 13% of drugs. So one of the things, and we talk to drug companies about this quite a lot, is so if you're developing a drug, you're running a trial, if you're not measuring the microbiome, then you have this huge black box that could be deactivating your drug. So some of the people that you're testing aren't even on your drug, really. I mean, they're putting it in their mouth, but it's getting changed to something else. They're not, so you've got this big black box. And so one of the things that, I'm a big proponent of is if you're testing drugs, you should absolutely be monitoring the microbiome and understanding, are those drugs being deactivated by the microbiome? Because you could fail the trial, not because of the drug not being good, but especially if you're doing it, maybe do it in a part of the country where the kind of bacteria that eat that drug are really prominent. And you have no idea, and how many things could that have happened to? It is really a fascinating question.
Yeah. Well, so you just mentioned another thing that's interesting. You said in a certain part of the country where that bacteria is prominent. So does the microbiome composition of people systematically vary from region to region?
Yeah, it does. And there are commonalities, of course, not like it's 100% different or something, but it varies a lot. They did a study. There's a Nature Medicine paper from 2018 out of China. And in that case, what they did is they took signatures that they would learn from the microbiome in one province, and associated with health, like this is a healthy microbiome, this is unhealthy. And they would, the machine learn these things. And then you would apply it in the next province over. And what happened is that the accuracy of that went down substantially because of this variation between microbiomes and where they are in different places and in different people. So a couple of things, though, that are quite interesting. So we wrote ourselves a Nature Biotechnology paper in 2019. And what we were able to show is that you could capture about 50% of the variance in the diversity of the microbiome that would filter down into information in about 40 metabolites that you would see in the blood. So one aspect is that you've got this massive diversity in the species, but it does funnel down to a much more manageable kind of number of things to look at in terms of what do you find in the blood that are products of the metabolism that's going on in the microbiome. And it's co-metabolism, right? It's happening both in our bodies and they're much like the TMAO example. So you can see that. And then a second item that we did, we published a paper in Nature Metabolism earlier this year, where we monitored, and I think it was in close to about 10,000 people, microbiomes as you get older. And so as we did that, what we found was that if people stay healthy, your microbiome actually becomes increasingly unique, meaning that it looks less like anyone else's microbiome. All right, so everyone's microbiome, if you stay healthy, it goes away if you get on a bunch of drugs or if you get hospitalized or things like that. But if you don't, if you stay healthy, your microbiome's kind of going along your life with you, and it starts looking more like your microbiome than anybody else's, and they kind of diverge a little bit. But even as that divergence in the microbiome species is happening, we looked at the metabolites that were associated, again, as they appeared in the blood, and you saw certain commonalities of the kind of things that would change through the course of life. And there was a commonality to that, even though the species themselves change. So what this means is that there's a lot that you can delve in on by trying to understand the reflections of the microbiome in the host, right? In the person, like what do we see that's coming out of that that's good and that's bad? And, that's why we incorporate a lot in our testing predictions about, what are the metabolic effects that we expect to see given the species and the gene content, that we measure out of your microbiome, for example.
So basically, the microbiome's huge, millions and millions, billions of cells of bacteria and other things living inside of our bodies, on our bodies. There's many different species and types of microbes that'll be part of that microbiome. The exact composition of that whole population could be relatively similar in me and you. It could be relatively different. But depending on what that full population looks like, what that full microbiome looks like, that's going to mean that certain nutrients, certain metabolites either are in my body or are not in my body as a consequence of it.
Or at different levels, right?
Or at different levels.
It's not an on-off necessarily, right? It could be different levels.
So this is another area where, it sounds like you really can't know unless you actually measure it. So how easy is this to do these days?
So this is super easy. There's a number of tests out there. Ours is out there as well, the Thorne gut health test. And you can get, and what we do is what's called metagenomic sequencing. So this means you're not only getting the species, you're actually measuring all of the gene content. So you can get that. And then what that allows you to do are calculations about what kind of metabolisms do we see represented in that microbiome. And you can relate that back to a number of conditions. We actually did a trial on personalized recommendations from the gut health test for people that were suffering from irritable bowel syndrome, which we published in Personalized Medicine 2 years ago. I think it was 2019 or 2020. And when we looked at that, what we were able to see was that you could get very significant amelioration of symptoms by following personalized recommendations. These were lifestyle and supplement type things, not drugs.
Mm-hmm.
But basically, you could take natural approaches, you could modify the microbiome, and you could get rid of a lot of the symptoms of IBS in this case.
So it sounds like the microbiome is relatively easily modifiable. And I'm interested in how easy it is in terms of like physical exercise versus diet versus like probiotics.
Yeah. So you can definitely modify it. It's more modifiable by prebiotics than it is by probiotics, but I'll give a little bit of a caveat. So one product that we've got, the Prebiotic Plus, we call it, but the reason that, and it's basically a two-dimensional printed disc.
Mm-hmm.
And what it has on there are three things. And this is one of the issues, because oftentimes if you just take a probiotic, oftentimes it won't change your microbiome. And the reason for that is that the species that are existing that are there already in your gut, for example, they're there because they competed to be there. Like they're good at doing what they're doing and surviving there. And so you introduce a new species and it can be hard.
Mm-hmm.
So one of the things that we did is we actually, in this product, we incorporate what are called bacteriophages. And so if you take a bacteriophage, then basically what you can do.
And that's a virus.
Yeah, and it kills bacteria. It's a virus that kills specific bacteria. So it's aimed at bad classes of bacteria, to use a simple term. So what you're doing there is you're creating an ecological niche. So you're creating some space. Then you put in a probiotic, right? The good species that you want to get in there. And then incorporated on the same disc are prebiotics. So the fibers that we know that the probiotics most like to eat. So what you're doing is you're creating a space. You're adding in what you want to now fill that niche, because now there's a little bit of space for them to compete. And then you give them the stuff they grow on best to help them outcompete and change your microbiome in that way. So those are the kind of things that we think about. So you can get a microbiome to change, but you do have to you've got to create some space. Or prebiotics, just because you are changing the structure of the food sources, right? That can have a more persistent effect usually than the probiotic. But that combination approach is what I really
see. I see. So if you were trying to like grow a garden in your backyard and you wanted roses growing in it, you could just sort of blindly throw seeds in the yard, but it might not grow because you've got a bunch of weeds and other stuff back there. But if you pull out the weeds and make sure the soil's right for that plant or that flower, then it can grow. Is that the basic idea?
Perfect analogy. Yep. That's exactly right.
So that's the difference between prebiotic and probiotic. And probiotic is like you're adding something, and prebiotic is like you are preemptively creating the right environment.
That's exactly right. The probiotic is essentially, the flowers you're going to plant. The probiotics, the flowers you're going to plant. The prebiotic is the fertilizer that is optimized for the plant that you put in, basically the food that it most likes.
So you said that you guys at Thorne, you have one of these products. What can you just physically describe it and how much it costs and how you actually use it?
Yeah, sure. So we've actually got a whole suite of products that are associated with the microbiome test that get personalized recommendations for people. I think there's something like 40 or 50 different products that can be recommended depending on what the issue is. Mm-hmm. But the more general ones we do. So one is with a brand called Effusio, which actually I quite like these. So these are, and I actually take this every day myself, but it's a two-dimensional printed disc. So what you do is you take the disc and you just drop it in water. It can be hot or cold. I typically do it cold. You drop it in, it dissolves, and basically it's a beverage. It's quite good. Stephen Phipps, who's the guy who's our chief innovation officer, and he really makes all the products. But he was also a chef in his younger days, so it's kind of funny. So he's like a chemist, but he also has a good sense of some of these things. Really good guy. But basically, as you drop that in, and so it just becomes it's a berry flavored drink. I forget the exact cost. I think it's something like $40 for a month's supply, something like that. And it's just something that you drink. So that's one. And that's the one I mentioned that has the bacteriophages, the probiotics and the prebiotics kind of all in one. So that's one element. We've also got other products, one called FiberMend and EnteroMend, which essentially have a bunch of nutrients that are good for your microbiome and adds fiber